So again, I welcome to everyone on this great day where we meet about this molecular modeling procedure. Like, this is a technique which is going to help chemists, biochemists, physicists, biophysicists, and people from diverse field to enhance their skills about this. So today, me and myself, Sabahuddin and Haris, are here on your disposal. And if you have any question, please feel free to write in the chat. And after the session, we will also have some question answer session. But you can just post your question in between in the chat. So let's move ahead. And before we briefly introduce ourselves, I would, as our tradition from Aligarh Muslim University, I would like to request Mr. Safdari to kindly recite a verse from Holy Quran. Please, Haris.
Bismillahir Rahmanir Rahim. Alhamdulillahi Rabbil Alameen. Ar-Rahmanir Rahim. Maliki Yawmiddin. Iyyaka na'budu wa iyyaka nasta'in. Ihdinas siratal mustaqim. Siratal ladzina an'amta alayhim. غير المغضوب عليهم ولا الضالين.
Thank you, Haris bhai. And now with this, I would like you to please kindly introduce yourself, Haris bhai.
So I am Haris Safdari, and I am pursuing my PhD at University of Hamburg in Department of Chemistry, Institute of Molecular Biology and Biochemistry. Yeah.
Thank you. And myself, Sabahuddin Ahmed, and I am doing right now my postdoctoral. I am doing a postdoc in Forschung Center Munich, which is a research center in western state of Germany. And before that, I wrote my thesis on in computational biology at the Heinrich Heine University. It's in a city called Düsseldorf. It's also in the Western Germany. And before that, I worked for like 2 years at Central Drug Research Institute. Again, it was a computational biology project and in Lucknow, CDRI, you know. And I did my bachelor's in botany from Aligarh Muslim University, and I did my master's in bioinformatics from Jamia Millia Islamia. So this was a kind of brief introduction about us, and let's move ahead to what we have for today. Okay, yeah. So what we are going to learn today, so about the protein data bank. Maybe most of you or some of you already know this thing called PDB. And we will try to learn how we can visualize biomolecules using PyMOL. And then we will also quickly look into how we can make figures for publications. And of course, many of you know that publication is quite important part of a scientist's life. And then we will make a break of some minutes, depending on how far we are. And then we will look into the modeling of three-dimensional structure of protein. Like we will look into with the web server possibilities, and also like how we can model the protein locally. And during this modeling exercise, we will also look into how we can look into the template structures from the NCBI BLAST program, how we can identify the sequence, from where we can get the sequence, and so on. So let's go ahead. I hope you already look into the instructions which I sent around, and that is going to be helpful very soon. So why I talk about myoglobin now, because it's the first protein structure which we know, which was resolved in 3 dimensions, and it was solved in 1958 by John Kendrew and his co-workers. And it is commonly found in muscle cells, and that's why you see the meat color is red. Why it is red? Because of the myoglobin. And what is special about is that it stores the oxygen for muscles, which is going to help the muscles to work when it is needed. And this it does with the help of the heme group, and heme groups is some group which holds iron. So the oxygen is bound to this heme group. And so in the PDB, like when you look into the PDB, so there are several information which you find. So when you search in the PDB, you will get the ID, and this ID of myoglobin is 1 MBN. So every entry in the PDB has a unique 4-digit code, which helps a person to identify that particular protein. So it's easier to identify a protein. Like myoglobin may have different structures, but which structures are you interested in? That you can identify with 1 MBN code or PDB code. Like if you put this 1 MBN code there, you will identify the structure which we are going to talk about now. And then you will also find so many information which we can have a look when we open the PDB database there. So there you can find which method is there, what resolution of the structure is there, and so on. And what is interesting about PDB is there is a growth over time. And not only human, but bacteria and viruses and structures of a different type of organism is there in this database. And as you can see over the time, it's the X-ray and the structures which were resolved with the help of X-ray were of course increasing over the time. So I have this data until 2020. For sure, in 2021 there was something more. But you can also see slowly the crystal structures were also solved with the help of electron microscopy, which helped to look into the protein structures which were more complex. And so this is how the protein data bank is growing over the time. If you look at this database, protein data bank, you will, when you will open, you will find it something like this. And let's look into the protein data bank directly. Like that will be more helpful if I directly go to the website and show you how it is there. So let me share again. Where is the option? Yeah. So I hope you can see the screen, right? It will be nice if someone can say yes or no. You can see the screen of protein data bank.
Yes.
Yeah, okay, thank you very much. So we were talking about the myoglobin, right? So the code was 1 MBN, yeah? And then if we open this structure, so there are so many options. And so the first option here, you see the summary. The summary is like, so like a classification, what type of the protein is that, and if there is some mutation, and what date it was deposited, and who were the authors, and then the experimental data. So which method was used? It was electron microscopy, it was NMR, or it was X-ray diffraction method. And what was the resolution? And resolution is important here because the higher the number, lower the resolution. And then on the right side, we have this WWPDB validation is, and there you see the validation scores. And of course, we want these bars not in the red region, but in the blue region. So this is how we, this is how we also discriminate the different structures which we should consider in our computational study. So if it is in blue, it's better. So you see there are 3.3% Ramachandran outliers, 15.2% of sidechain outliers, and clash score is 54, and so on. So better, it's closer to the blue, it will get lower, and that's how it's this validation section is important for us. And then on the top, we have so many options here to download, and here you can download the FASTA sequence. And I want to remind you that this FASTA sequence is not the FASTA sequence of the protein, but the FASTA sequence of the protein which was resolved, that the crystal structure is of about. Like sometimes it happens that when you are doing the crystallography procedure, the crystals are not forming, and then you need to do some kind of mutation, for example, thermostabilizing mutations, and then when the sequence changes. So the FASTA sequence is not necessarily here the same as the wild type. So this you should remember. And then you can download the file in the PDB format and whatever you would like to download. And this is what you have here. And then you learn about the information called micromolecular content, atom count, and everything is there. And so again, here you look into the structures, which chain it is, how long is the sequence, and then what is important here to look into, was there any mutation? If there is some mutation, then you have to look carefully what mutation was there, what substitution was there. And so you can scroll down, and there are so many things you can look into, what ligands were included in the structure, and so on. So again, this is the same validation report, but upper side was a summary. It's more detailed things. So this is how the protein data bank look into. So so if you don't know the code, you can also look into directly. For example, one of the proteins which we will look into is oxytocin receptor. So this is the protein on which I am working right now in my postdoctoral position here. And that's why I talk about this. So many of you know about this oxytocin receptor, which is kind of very important for late pregnancy. And there are so many functions, and not only in the pregnancy, it is also important for in a nervous system, a central nervous system. It has role to play, for example, social bonding and anxiety, and so on. There is so many function of this receptor. So I meant to say, like, instead of, if you don't know the code, you can just type whatever you want to search here, anything, what else. Yeah, so we search, and like, for example, again, I can search here myosin, and you can see the structures of myosin, and so on. So this is it. So we will come back again to our slides. Okay. And where is the slide? Yeah. Yeah, so here it is. So we were here. So we already looked into the protein data bank now. And what is our next goal is, so I didn't told you to install one of the files, like to read into the PDB files, but I can show you now how it looks like from inside. So generally on the top, you have the information in the PDB file. When you open in, let's say, Word file, it will not open that properly, but there are other programs in which normal text file, you can open it, and then you will see that it has a few very important information. For example, on the top is general information, like header, it says oxygen transport and what is it about, and then specific information of method which was used to identify this, so an NMR study and how it was done. And then the third thing is atom types and positions, coordinates, and so on. And as you can see here, so atoms, so the first column is about atom, and then atom number, and then atom type, and then third, which residues correspond this atom correspond to, and then other three over here correspond to what are their XYZ coordinates, and so on. And this head atom means the ligand. For example, the heme atom, or this iron, for example. Now I can switch on the. I can, yeah, okay. Yeah, I'm sorry. Yeah, so I wanted to show you this, for example, this Fe, this is also at heteroatom, it's iron, and then you see all the coordinates here and so on. And after this, let's move to the next slide. And I will shortly pause the recording for a while, and then we will get back. So if you have any questions, just raise your hand or just ask right now. Yes, Subhana.
Sir, sorry, I could not follow the last part where you were labeling and changing the amino acids.
That thing.
Yes.
Okay, so I can just show you again. So this is the PyMOL we opened. Yeah.
Yes.
Yeah, you are able to follow my screen, right?
Yes.
And you go to wizard, and then you go to mutagenesis, and then go to protein. Okay, and then you decide on what residue you want in the last. You want alanine, arginine, cysteine, histidine. So let's say I want proline. Okay, I select proline, and then which residue you want. For example, let's say I want this one. And then there are different possibilities. You can just select which one, and then select apply. And then this is turned into proline. But to do the mutagenesis, or let's say substitution, I will not recommend PyMOL, but I show you this because I wanted to show that PyMOL can do so many things. So Mr. Saftari just said in chat recently that you can use you can use Chimera, and Chimera is stronger than PyMOL, but PyMOL it's like quite easy to it's for beginners. It is quite easy. So that's why I wanted to show you this PyMOL. I wanted to do this workshop with PyMOL, but next time we can also do it Chimera. For this kind of substitutions, I recommend fold X, which I wrote in the chat. That's kind of very useful, because it also, when you do this substitution, it also gives you the energetic terms which you can report. For example, the difference between the wild type and mutant, what will be the difference in energy, and then that corresponds to the stability of the protein. Yes, Sidra, you have a question.
So I was just wondering if you can share the presentation after you complete all the classes so that we can have a look and go through it while we are practicing.
Yeah, I will try to look into when it is possible. I can share the presentation for sure. For the people who participated in this, I can just write an email. So I don't know. So how we have a record of who joined today. So it will be nice if you can all write yes. So I know the names who joined today's session. So just write in the chat yes, like attendance. So I can send you the slides. So you have more questions? Yes, Ifra and Sana, either of you.
So this when you changed, like you had to polar contacts. So how did you create that object? Because you didn't give the command right in the command box. I couldn't follow that.
Like when that's also I missed, like when this happened.
Yes, like how did you change like to the polar contacts? You created an object by the polar
Okay.
contacts. Yeah.
Okay, so let me share the screen again. Because I share the screen. Okay, so like this. Yeah. And so this is a complex, right? 1 MBN. You just go to the action, and then you go to preset, and then you decide what kind of preset setting of PyMOL you want to use. So this was the option which I selected, preset ligands, and this was there. Was this you meant?
Yes, this is what I meant, because
There
I
are so
couldn't
many.
like, yes, I was trying to find it and I couldn't like
No,
where
there are
did it
so many
come from.
options. You have to go to action and then
Okay.
preset, and there are so many options, like you can, maybe there is protein interface also, and like there are so many options. So I like to do it with the ligands, and then what I do is I create I just make the residues which I want to show in sticks, and then I show the cartoon as well, like more fancy, making the figures more fancy for reviewers of the publication, and then just save the image.
Yes, got
Yes.
it. Okay, yes, thank you.
So yeah, there was one more raised hand.
Sir, can we add amino acid sequence rather than substitution?
Sorry, again, please.
Can we add some amino acid sequences rather than substitution?
That I never tried. That is good question, but I never tried that. I never tried. I don't think there is option, but maybe there is option. I never needed it, so I don't know, honestly.
Okay.
But in this software called Chimera, you can rename the sequence numbers, and there are so many options more than PyMOL. You can do short minimization and so on. I can write in the chat so you know which software I'm in. There is the chat box. Where is the chat box? Chimera and Chimera X, right? Chimera X. So there is more question by Sana, right? Sana?
No, sir. Only that was my question. I wanted to know that if we can add some sequences or not.
I have a question.
Okay, then you can lower the hand. Sidra, then your question, please. Ashi,
I
you.
just want to know, like, do you guys keep organizing such kind of bioinformatic conferences in future also for like this is like for beginners, and eventually will the level will keep increasing so that we can learn actually
Yes, indeed, that's the plan we
molecular,
want
like
to.
for example molecular dynamic simulations are very much uh,
In demand.
in demand these days. Yeah.
In demand.
So
So wait, just a minute. So Ashi, like, you mentioned that is it for over for today? So I think it's like audience is like kind of tired. So we will continue about the next things from tomorrow onwards. So if you have some appointment or you have some plans, you can also leave. Coming back to you, Sidra, sorry for breaking the conversation. So yes, of course, we plan to do the molecular dynamic simulation things, and maybe I'll take that as well as there are other people as also who can take this session, molecular dynamics. In fact, my PhD was solely based on the molecular dynamics simulation, and before coming to Germany during my project, I also did molecular dynamics simulation. It's kind of very helpful. So in the problem with the molecular docking is that it gives you just a idea about a snapshot. It's like a snapshot. It does not give you idea about the how the ligand or how
interacting with the
yeah, exactly. So what is missing there is what is happening on the time
water
scale.
molecules are missing.
Exactly. So what is missing is on the time scale in the docking. So when we do the molecular dynamics simulation, we learn things in the time scale, and that's why molecular dynamics simulation is very important right now, or for coming future is quite important. And then if you want to learn molecular dynamics simulation on your own, I can recommend you very nice website. So you can already start looking into, let me find it. And when I was after my master's, I wanted to learn about this, and this tutorial was kind of very useful. Even lab tutorial, no, it's it was from yeah, I found it. So depending on what you want to do, membrane simulation or protein in protein in just enzyme in water, you can just choose which one you want. So I will suggest, like, if you want, when you start with very basic, and this one is the very basic one. So they provide you with the input files and everything. You just follow the tutorials, and it's written in very, very easy language. you will learn in very short time. And to learn about the basics, I can recommend a very good book which is available at a very low cost in Daryaganj or in India. Andrew Leach, Molecular Modeling. Because I ordered when I joined my PhD, my boss said, "What is the book which you follow?" And I said, "I don't have a book." Then he was laughing, and then I ordered the book, and this book came to me from India.
Okay.
Andrew Leach is the name of author, and it is very good book if you want to learn the basics of the molecular modeling or free energy and so on. So it's very cheap. It's like 1,000 rupees or so, or less than that, and then just follow this book to learn, and this tutorial.
As far as I know, I think the MD simulation cannot be controlled by small computers because it requires a lot more power and supercomputers are needed for it. So we need a server to connect. Like
Wait. So when I was unemployed, I didn't had any job. So I had the one collaboration from biochemistry department of AMU, if you know Imrana ma'am. So there was a collaboration with her lab, and I did some MD simulation, and also Masood sir was there. So one collaboration was with her. So first publication which I published was I did on my laptop. You can imagine, in 2011, I brought that laptop. It was first generation i3, and I published this manuscript in PLOS when we shared the first authors, and it is just 2 nanoseconds. The problem nowadays, people want to see at least 10 nanoseconds, but it doesn't make sense. If I achieve this 2 nanosecond in 4 days, you can achieve 10 nanosecond in 1 month with this slow computer. But right now the computers are way faster. Your laptop, I mean, your laptop is way faster than what I had in 2014 or 13. And you can for sure do longer simulation, and the speed or the time depends on the number of atoms in the system. You know what I mean?
yeah. Right.
So if you have a membrane protein, if you have a protein with dimers, primers, tetramers, then it has a large number of atoms, and then it will take time. And so for example, if you have a protein with 300 amino acid, it takes 2 days, for example. If it is 1,000 amino acid long residues, it will require more water molecules, more atoms, and it will take more time. So in my PhD, I had a membrane protein, which was a dimer, and it was taking on the local cluster, which is better than what we have at home. And it was taking 1 month time to finish 100 nanosecond, okay? And when I did the same thing on the supercomputer, because here I'm working at the supercomputer during my PhD also, and in my postdoc, I use the supercomputer, and in that it takes just 1 day to finish 100 nanosecond. So the computer makes difference, but it is not that you cannot do at your end.
Also, I have heard that these days you can dock to find drugs for your like suppose you are interested in finding drugs against a particular disease, so you have to select drugs from a reservoir of drug bank, and then you have to see by docking like which of the drugs are suitable, and then you can narrow your drug choice. So it's also very much in a hot topic, and you need to like narrow down drugs from a repertoire of the drugs. So that is
Of course,
also
of
something
course,
that's very much in boom. So that is
of
also
course.
interesting. Yeah.
Exactly, but that's not a hot topic. It's like old hot topic. It was in the past. It was in the past when like 6-7 years before it was like hot topic. And the paper which I shared in the chat right now, there we identified like there were several molecules which I did a screening by molecular docking. And before I did the molecular docking, I had to filter out which one we need, and there are different ways how we can filter which molecules is more useful. For example, there is this Lipinski rule of 5, if they follow or not, if there are peptidic molecules or they are non-peptidic molecules. This may help you to decide. And there is this thing called like pharmacophore based screening of molecules from database. And that is some other topic which we can also discuss other time. I don't plan it to cover it tomorrow with the docking session, because the docking is itself, it's a time taking thing. But in near future we can do this workshop called like virtual screening of molecules, where we can work with like, actually this thing is something like you do together, like I show you and then you will just see and then you'll forget. It's something you do together, like we did PyMOL together. But with the virtual screening, you need a database, you need more installation, and then we want to be sure that everything is like perfect. We can for sure organize something like that in near future.
Okay, thank you.
So something more, if anyone has some more questions or comments. So it's already like kind of 10 in the night in India. So if we don't have any further questions, we can stop the session here, and then I hopeful we will see or hear each other tomorrow again. And yeah, I hope that we will finish tomorrow. We don't need a third day. I hope so. I really hope so. So thank you very much for everything, but don't close the Zoom until you save the chat. So when you are closing your Zoom, remember to save the chat or copy the whole chat somewhere, so you have the command. And the document which I will share with you, I think they also have this kind of list of commands. If so don't worry. So like if you missed some points, like if you you are always free to write to me or Mr. Safdari. So like it's always, we are always there to help you. Like individually we can help you. It's just that you are willing to learn. We are happy to share the knowledge to you, everyone. So if you are not sure about something, if you want to ask something, just write to us an email. Everyone knows my email address. I'll be happy to help. So thank you very much. And it was pleasure to hold this workshop for tomorrow. See you then. So, my email address. Yeah, okay. So Mr. Safdari's email address is in the same email which I sent to you, Ahmad Khan. Just have a look at the email. He is copied in the email address. Yeah, okay. Everything is clear. So see you tomorrow. Allah Hafiz. Bye bye. Have a nice rest of the evening.
Allah Hafiz. Thank you.
Allah Hafiz.